This protocol helps to understand the spatial and temporal recruitment of DNA damage repair proteins, taking into consideration the cell cycle phase. For cell cycle discrimination, we use fluorescent protein tagged pCNA as a marker of S phase, which circumvents artifacts introduced by other cell cycle synchronization methods. This, combined with laser-based micro-irradiation, gives unparalleled spatiotemporal resolution to DNA damage repair protein kinetics. Successful and routine utilization of our protocol builds on basic knowledge of confocal imaging.
"24 hours before the micro-irradiation, plate a total of 8 by 10 to the 4th cells and 500 microliters to 1 milliliter of media on a 4-well chambered cover glass. 1 hour before the microirradiation, exchange the regular growth medium with imaging medium containing either olaparib or a vehicle control such as DMSO. At least 4 hours before imaging, turn on the environmental chamber and the microscope components. Switch on the heating, carbon dioxide supply, and the humidity regulator. Initialize light sources along with the laser lines at least 1 hour before transferring the cells to the microscope. Oil the microscope objective and place the chambered cover glass under the microscope. To select S-phase cells in an asynchronous population, look through the ocular for the unique localization pattern of the M-plum tagged pCNA in S-phase and select a field of view that